Hello everyone, welcome back to my channel, Igcse Biology Help. In today's video, I'm going to be solving the paper 4 extended for the October-November 2023, paper 4, variant 2. I have already posted variant 1 on my channel, make sure to check it out. So let's start solving. Question number 1 says, red blood cells are specialized cells that transport oxygen. State the substance in red blood cells that combines with oxygen. This is actually hemoglobin. B says, state the name of the component of blood that promotes blood clotting. This is the platelets. C says, some students investigated the effect of immersing red blood cells in different concentrations of salt solution. So this is about diffusion. They measured the diameters of samples of red blood cells and calculated the mean. Then they immersed each red blood cell sample in a different concentration of salt solution. After 2 minutes, they measured and calculated the mean of the samples again. Table 1.1 shows the results. So as we can see that as the percentage of concentration of the salt solution has increased, the mean diameter of the red blood cells, they have decreased. As you can see over here, there was a very low concentration of the salt solution. There was a bursting of the cells, and when the concentration of the salt it started to increase, the cells, their diameters also have decreased. So there is basically an inversely proportional relationship over here. So let's see question I. Calculate the percentage increase in the mean diameter of red blood cells that were immersed in the 0.8 salt solution. Give your answer to two significant figures. Okay, so first of all, if you want to calculate any percentage change, then you have to get the you have to get the new value, and you have to minus it from the old value. And I'm going to be solving right now to show you an example, and then you're going to divide it over the old, and you're going to multiply by 100. So we have the new value over here. They're asking about percentage increase in the diameter of the cells that were immersed in 0.8 salt solution. So first of all, it increased from 7.5 to 8.2. So we have to calculate how much it changed. So you're going to be, this is the new value and this is the old value, because it was like that in the beginning, and then it changed to 8.2. And also guys, the new value doesn't have to be larger than the old value, it can be smaller. So this is how you calculate. You're going to do 8.2 minus 7.5, which was the initial diameter, and you're going to divide it again over 7.5 and make sure to multiply by 100 so that you get percentage increase or decrease. This is going to give you 9.333 and recurring decimal. So you're going to only write 9.3 because they want the two significant figures. Double I, explain the results for the red blood cells that were immersed in the 1.8% salt solution. So the salt solution, as I said, it will have lower water potential than the red blood cells. So the diameter will decrease because the water, there will be more water that will leave the cell by osmosis. So it will move from higher water potential to lower water potential through partially permeable membrane. So this is basically what you say. So as we can see over here, there is lower water potential. So the cells, they will move into the cell because there is lower water potential as there is higher salt solution. So the diameters of the cell, it will actually increase. Triple I, state why there was no change in the mean diameters of the red blood cells immersed in the 0.9% salt solution. Water potential of the red blood cells is the same as the salt solution, so no water potential gradient. So basically over here there was 0.9 and there was no change. This is because the solution over here, if we read, it had the same concentration as the water as well, so there's no actually change. So this is the answer. G says state why red blood cells burst when immersed in pure water, but plant cells do not. Red blood cells do not have a strong cell wall. So we know that the red blood cells, they don't even have a cell wall, so you can also say that. So plant cells, they actually have cell walls and they are made out of cellulose, so that's why they don't burst. Red blood cells will burst if there is too much water because it can't handle pressure as there is no cell wall. State 2, uses of water in a plant. You can say it can be used as a solvent, or it can be used in translocation, or you can say it is a medium for chemical reactions. That means that water is used in chemical reactions so that they can occur. So if there is no water, the plant will be dehydrated and there will be no chemical reactions or respiration taking place, so it will die. So it's very important that the plant has water supply. Figure 2.1 is a photograph of some leaves of a water lily, which is a hydrophyte. The water lily has adaptive features that are found in many different hydrophytes. A. Describe what is meant by an adaptive feature. It is an inherited structural feature that helps an organism to survive or reproduce in its environment. So this is a definition that you should have memorized. It's in the syllabus, and I have made a video about all the definitions that you have to revise and memorize, because some questions you have to write exactly as the definition that is in the syllabus. So make sure to memorize all of these. This is not stuff you have to understand, just memorize it and write it if they ask about it. B says, figure 2.2 is a photomicrograph of a cross section of a part of a water lily leaf. State the names of the parts labeled A, B, and C in figure 2.2. So A, this is actually the palisade mesophyll layer, and B over here, this one, it's actually the spongy mesophyll because it has a lot of air spaces. As we can see, this is how you know this is a spongy mesophyll layer. And C over here, they are pointing towards this empty area. So this is actually the air spaces that are present within the spongy mesophyll layer. I says explain how part C in figure 2.2 adapts the hydrophyte for its environment. So large air spaces, they actually help plants like water lily to float on the water and adapt for its environment and attract more sunlight. This is because the large air spaces, they will reduce the density or the weight of the leaf itself. So it will help the leaf to actually float on the surface of the water, and it will be like it will be exposed to more sunlight. So this is also a feature, and it allows it to float. So it allows it to be more accessible to light, as I said, and carbon dioxide gas exchange for photosynthesis. So this is very important that the plant floats so that it is exposed to more air and more light and carbon dioxide. A scientist calculated the mean number of stomata per millimeter square in the upper and lower epidermis in the tomato plants and water lily plants. Tomato plants are a type of terrestrial plant. Table 2.1 shows the results. So over here we can see that the tomato has a lower number of stomata in the upper epidermis. Water lily has a lot of stomata in the upper epidermis, and in the lower epidermis it has zero, unlike the tomato, which has a lot of stomata on the lower epidermis. So we have to compare and explain the differences in the mean number of stomata in a tomato plant and in a water lily plant. So in the previous question, I have explained that the water lily, it floats on the water. So this means that all of its cells will be concentrated on the upper epidermis, because this is where mainly all the photosynthesis and all the chemical reactions take place, and because it floats on the water. So the other side, which is the lower side of the water lily, it is zero because it is already in the water. So there is no like all of the cells are exposed up in the sun, and at the bottom there's no sunlight reaching or anything. So in the tomato it is the opposite, because it is all of the action takes place in the soil, germination and everything, it takes place in the soil. So all of the stomata have to be at the lower epidermis. So they're saying explain and compare. So first of all, you're going to say one comparison and you're going to explain why that is. So let's see, tomato has more stomata on the lower epidermis than its upper epidermis, as it's not facing the sun. As I explained to you guys, it's not facing the sun. So as I said, all of the action is taking place in the soil, not in the sunlight or like upwards, because it is in the soil already. Water lily has stomata only on upper epidermis, no stomata on lower epidermis, and you have to explain why that is. So you're going to say, as only upper surface is exposed to air. 3. Water lily has more stomata on upper epidermis than tomato, as water lily floats on water and its upper surface is more exposed than the tomato to air. So it's basically the same point, but you have to say the point. So you're saying here different comparisons and you're saying why that might be. So this is basically explanation. Double I says, state the name of the cells that control the opening and closing of stomata. These are the guard cells. Question number 3, figure 3.1 is a diagram of the junction between two neurons in a healthy person. Figure 3.2 is a diagram of the junction between the same two neurons in a person who has Parkinson's disease. This disease affects the nervous system. So A says identify the parts labeled X and Y in figure 3.2. So over here, this is actually the vesicle membrane over here. If they have made the arrow inside like that, then you will say vesicle only, but they have basically pointed at this line. So this is very similar to the questions in the plant cells, where they put an arrow in the cell membrane and another one in the cell wall. So be very careful in these questions. Y says synaptic gap. This is the synaptic gap over here. So as we can see here, let's analyze a little bit of these figures. Figure 3.1, we can see that there's very little in figure 3.2 we can see that there is lower amounts of vesicles that are in the neurotransmitter, but over here we can see that in figure 3.1 there is a large amounts of the vesicles, and there is a large amount of neurotransmitters over here than over here. As we can see, there is a difference. So let's see the rest of the questions. B. Parkinson's disease affects neurons in the brain that are responsible for movement. Using the information in figure 3.1 and figure 3.2, suggest and explain the effect of Parkinson's disease on a person's movement. So we can see that there is going to be slower movement because, as I said, there is lower amount of neurotransmitters over here, and there is lower amounts of vesicles. So there is lower amount of overall, there is lower connection between the receptor proteins and the neurotransmitters. So let's see. There are slower reflexes and fewer vesicles. There is fewer neurotransmitters in vesicle X, the fewer neurotransmitters released when an impulse arrives, which leads to fewer neurotransmitters diffusing across the synaptic gap. So over here in the photo, it's very clear there is lower amounts of these little cells. These little cells are actually called the neurotransmitters. These are in lower quantity over here, so there will be lower binding of these little cells with the receptor proteins, so there is lower reflexes overall because there is lower binding. C says describe two ways nervous control differs from hormonal control. So of course hormones are much slower than the nervous system. So but there's also another point. The hormonal control, it is more long-lived effect. This means that the hormonal control will stay, its effect may stay for a few days, for example, but the neurotransmitters and the nerves, they will only stay for maybe 10 minutes or 2 minutes. These are very short-lived, and the transmission is through nerves for the nervous control, and it's faster than hormonal control. DI says the shape of the receptor proteins shown in figure 3.1 and figure 3.2 is important for their function. Explain how the shape of the receptor proteins is determined. So, of course, they are determined from the sequence of bases in the DNA or the mRNA that is transporting the DNA sequence, and it can also be difference in the sequence of the amino acids. And sequence of amino acids will determine the shape of receptor proteins. Double I says cell membranes also contain protein carriers. Describe the role of protein carriers. So they move ions in or out of the cells. These are actually using energy from active transport by respiration. So their movement is against concentration gradient, moving from low to high concentration. 4. A scientist investigated the effect of temperature on the rate of photosynthesis in one species of plant. Photosynthesis involved in enzyme controlled reactions. Discs were cut from a leaf and kept at different temperatures. The total surface area of the disc was kept the same for each temperature. The volume of oxygen that was produced by the leaf disc was measured and used to estimate the rate of photosynthesis. The results are shown in figure 4.1. So we can see that as the temperature increases over here, the rate of photosynthesis, it will increase until this point over here at 40 degrees, and then it will decrease. So they're saying to identify the optimum temperature for photosynthesis. So the optimum temperature is where there was the most or the highest rate of photosynthesis. This was actually over here at the temperature 40. So you're going to write that down. B says explain the results shown in figure 4.1. So this is a 6-mark question. You have to explain everything from before 40 and after 40 and during 40. So first of all, before 40 there was increasing temperature, and this increases the kinetic energy of molecules. So we know when there is increasing temperature, there will be increase in the kinetic energy, and this will increase the frequency of collisions, leading to more enzyme substrate complexes forming. This is because there is higher energy and there is faster movement, so the cells will meet each other more frequently, and there will be higher amounts and higher rate of enzyme substrate complexes forming. This will lead to an increase in the release of oxygen or glucose products. The temperature is the limiting factor at 40, because after that the rate will decrease, because um, there is higher temperature, and there will be denaturation occurring, because the rate will continue to decrease until the shape of the enzyme substrate complex it will change. So increasing the temperature increases or changes the shape of the active site of the enzyme. This will lead to lower amount of enzyme substrate complexes forming, because they can no longer bind, because the active site of the enzyme is changing and the cells they cannot bind anymore. This will lead to denaturation, in which substrate no longer complementary to active site and can no longer fit into active sites. There is going to be fewer enzyme substrate complexes forming. So this is basically explanation of all of the graph and why all of this happened. C says carbon dioxide was supplied in excess at each temperature during the investigation. Explain why. So carbon dioxide was supplied in excess so that it does not become a limiting factor, so that the plants can have sufficient amount of carbon dioxide, and that the result we can ensure that the result is only caused by changes in the temperature. So if you have to control the environment of the plant, so that you are testing only one variable at a time. You don't test, for example, the changes of carbon dioxide. And also you can say for more accurate results, so that there is no limiting factors other than the temperature. Suggest why not all of the oxygen produced by the leaf is released, because some of the air will be trapped inside of the air spaces, or some may be used in other chemical reactions such as respiration, so that the plant itself can continue to live. E says describe the role of chlorophyll in photosynthesis. It converts light energy into chemical energy for the synthesis of glucose or the production of glucose. 5a, describe two ways in which the circulatory system of a fish is different than the circulatory system of a mammal. So the fish has single circulation, which means the heart, it will pump the blood once only, not twice. So the blood will flow until like the end, until the lungs. It will not be re-oxygenated like the double singulation. So there is also no septum in the heart, so the oxygenated and deoxygenated blood, they will combine to each other, and this will cause lower amounts of oxygen being released. B says, explain the advantages of a double circulatory system. There will be it will prevent the mixing of the oxygenated and deoxygenated blood, and the blood will be able to flow at a higher pressure. This is because it is pumped twice, so the pressure, it is not as the blood flows, the pressure is decreasing, as we all know. But when you pump it again in the double circulation, it will be able to still flow at high pressure, and it's pumped faster because there is, of course, double pumping. So there is also fast supply of nutrients as there is fast supply of blood, and there will be also fast removal of waste and carbon dioxide and urea and lactic acid, because again there is fast supply of blood. C, figure 5.1 shows part of the circulatory system of a mammal. So it is actually a double circulation. I say, state the letter from figure 5.1 that identifies where these processes occur. So absorption of the products of digestion into the blood, this is at C over here, which takes place in the small intestine. So excretion of carbon dioxide from the body, it takes place from exhalation. So this is at the mouth or from the nose. Formation of urine, this is basically at the kidneys, and production of bile, it is at the liver, which is at B over here. Identify the name of the blood vessel labeled X in figure 5.1. It is over here, which is near the liver, which is going to be called the hepatic portal vein. Describe the role of the liver in excretion. It is used for deamination, which is removal of nitrogen containing part of amino acids, and it also is used for removing excess amino acids from the blood and formation of urea and breakdown or respiration of lactic acid. 6a, state the names of two hormones released by the ovaries. They are estrogen and progesterone. During pregnancy, antibodies are acquired by the fetus from the mother. State the organ that the antibodies cross to reach the fetus. This is actually the placenta. C, a baby was breastfed for 6 months. The concentration of antibodies in the baby's blood obtained from the breast milk and the concentration of antibodies made by the baby itself were measured. So over here we can see that the antibodies produced by the baby, it was increasing, and also when the breastfeeding stopped, the concentration of the antibodies in the baby's blood was constantly dropping, but it was producing for itself at the same time. So let's see the question. Complete the sentences to describe the changes in the antibody concentration in the baby. After birth, the concentration of the antibodies acquired from the mother decreases rapidly to 6 arbitrary units at which month? So it is actually over here at 6 months. It stopped to, the breastfeeding stopped, so the antibodies concentration was at 0 over here. So this is also when the breastfeeding stopped, and also the antibodies the baby made were increasing at the same time. So antibodies start being produced by the cells called lymphocytes in the baby immediately after birth. Total concentration of the antibodies in the baby from both sources was, you're going to calculate this question, so let's calculate it together at 4 months. So first of all, you're going to be getting the value of each one from the acquired antibodies and the produced antibodies. So first of all, at 4 months, the antibodies that came from the breastfeeding, you're going to draw a line until it meets over here like this. So from 4 months, basically, I'm going to draw a line like this upwards until it meets the black line, and then you're going to draw a line to see the value. So this is 10. So each square over here, if you divide 5 or 10 divided by 5, it's going to give you 2. So this is 2 and 4 and so on. So over here, this is going to be 18. So 18, this is the acquired. Okay, so let's see the produced antibodies. You're going to again draw a line starting from 4 until you meet the dotted line over here. So this is actually over here 38. So you're going to do 38 plus 18, and this is going to be your answer. So this is how you do it, because the lines don't meet at the same time, they are inversely proportional. So you're going to have to calculate each one in it at its own, and then you're going to you're going to do 38 plus 18, the acquired plus the produced, to get your answer. So it's going to give you 56 arbitrary units. The concentration of antibodies acquired from the mother and the concentration of antibodies produced by the baby are the same at which month? So you're going to see where they meet. So they met over here. This is the line where they met. This is where they were basically the same value. So you're going to write down this was during the 3rd month. So double I, describe the benefits of breastfeeding a baby for the first 6 months of life. This is going to provide the baby with passive immunity, which is going to provide sufficient amount of antibodies, because the baby cannot produce for its own antibodies because it's still young, and it will actually provide protection against or immunity from pathogens, and it will provide all the nutrients that are required by the baby, because it still cannot eat full food, so it can only drink milk. So it will have nutrients for the baby, and it will form connection between the baby and the mother as well. Triple I state 2 ways other than breastfeeding that a baby can acquire immunity. It can be through vaccination. It can be active immunity from infection. So the baby already got the infection, or the pathogen was inside of the baby's body, so it formed active immunity already. So there can be also passive immunity from the placenta, as we said in a previous question. They said that antibodies are acquired by the fetus from the mother, and we answered this question and we said that it was from placenta. So this is also another way other than breastfeeding that it can acquire immunity. D says, explain the importance of the shape of an antibody. So this is like the enzyme substrate question. You're going to say that each pathogen has antigens which are specific shape, and their shape is complementary to the antibody shape. So the antibodies will bind to the antigens, and the antibodies will mark the pathogens for destruction or destroy the pathogens because of the complementary shape. So they have to be complementary, and the shape has to be exactly like the antigen so that they can bind together. Antibodies are proteins. State the chemical elements present in all proteins that are also found in carbohydrates and fats. These are carbon, hydrogen, and oxygen. You cannot say nitrogen because nitrogen is only found in proteins. So this was the end of our exam. Thank you all for watching.